Hello and welcome to the 2.1 syllabus for this IB biology course. I'm going to first start off by defining some key functional groups which are highly relevant to metabolism. So firstly we have our hydroxyl group, which is a fairly simple structure seen in things like alcohols, and we know it is a polar covalent. Secondly, we have our carbonyl group, so we have a carbon double bonded to an oxygen, seen in things like hormones, most notably testosterone. It's also a polar covalent. A third functional group is the carboxyl functional group. We know that that's a carbon double bonded to an oxygen and then bonded to a hydroxyl group. This is seen in things like acids, for example, vinegar. And we know it's also a polar covalent. Next up, we have an amine group with a nitrogen bonded to two hydrogens. It's seen in things like DNA and amino acids, and it's also a polar covalent. Phosphate groups, seen in things like nucleic acid. To phosphate bounds to three oxygens, one of which is double bonded. Notably, these oxygens here are anions, being negatively charged. Phosphate's seen also in things like our energy sources, so ATP or ADP. Next up, we have methyl groups, seen in things like fatty acids most commonly. Methane's another example, and it's effectively just carbon bonded to three hydrogens, and then from that, the rest of the molecule. So it's important to talk about the actual types of formulas that we can have. We can have things like a displayed formula. So in the example of methane, this is its fully expanded formula, if you like. We also have things like structural. So in the case of a fatty acid, which I'll use for these two examples, we can see that we can note the structure. However, it's simplified in the sense that CH2, which would be written like that in the case of a fatty acid, connecting to more carbons, etc. It has been simplified just to be like that, and that's how it's represented. Finally, we have our skeletal structure, which is the most simplified structure. So this, in fact, represents your fatty acid with each line, if you like, representing this hydrocarbon chain, which is what I drew earlier. So that's your most simplified form. Now I'm going to move on and talk about carbon and why it's so essential in metabolism and in daily life. So it forms four bonds, which is important because it shows the molecules that it can form with or it can be a part of. It's involved with organic compounds, so that's with the presence of carbon and hydrogen. And we know it's seen in things like DNA, carbohydrates, enzymes, and much more structures. So our major carbon classes are things like carbohydrates, presence of carbon, hydrogen, and oxygen. And we know carbohydrates can be segmented into things like monosaccharide, disaccharide, or polysaccharides. So I just did some brief examples. We know a monosaccharide is something like glucose, disaccharide, lactose, of course, being made from glucose and galactose, and a polysaccharide being that chain of monosaccharides. So things like cellulose. Another important thing involving carbon is that of lipids. We know that's seen in the cell membrane. So things like fatty acids, and we know with the cell membrane that's going to be insoluble. If you remember, we have our phospholipid bilayer and we have our hydrophobic region, which is effectively this lipid. Next up, we have proteins, so things like amino acids and enzymes. And finally, we have nucleic acids, so DNA or nitrogenous bases, And we know these can be most notably defined through the presence of that phosphate group. So if we ever need to recognize one, that presence of the phosphate group will allow us to do so. An important structure to talk about is that of amino acid. Amino acid's basic structure is as follows, and one can note the presence of an amine group and a carboxyl group. Now, amino acids have the generic form of this. However, note that I've highlighted this R group. Now, this is known as the R variant, and we know that there are 20 amino acids that are seen, and this R group is what defines and makes each one unique. So I've put an image here of the 20 common types, and although it looks complicated, it can be summarized to the fact that each R group is unique and leads to each specific type of amino acid. So that's your basic structure, and it's important to know. And the R group is what defines each one individually. So some more molecular structures. Firstly, we have ribose and deoxyribose, and we know, of course, their relevance in things like RNA and DNA. Now, they go under the following molecular drawings, with ribose having a notable difference to deoxyribose in this particular bond. Deoxyribose, in essence, means that there's that D oxygen, so implying that there's less oxygen. And that is most definitely the case with a lack of oxygen at this particular bond compared to your typical ribose. So we know they go under the formula of C5H10O5, with deoxyribose having one less oxygen, so it's going to be O4. I've gotten some images here to illustrate each one separately. So obviously deoxyribose and ribose, and their structure is effectively what the image portrays. But yeah, really important to note that hydrogen and that hydroxyl group in the ribose. Moving on to glucose, there are two forms of glucose, both of which are seen differently. They have the same formula, being C6H12O6, and they go under the structure as follows. So there are effectively, we have 1, 2, 3, 4, 5 carbons in that structure, and then we have a carbon bonding to your fifth carbon. Now, a good way to memorize these drawings is to look at the presence of the hydrogen and that hydroxyl group bonding at each of those carbons. So we know in alpha glucose, the hydroxyl group is bonded at the bottom, whilst the hydrogen is bonded at the top to that carbon. Now, a way I remember is to look at something like beta glucose, which again has that same structure, but a notable difference in that first carbon. As you can see that they are effectively the opposite. We have the hydroxyl group on top. So the way I like to think about it is looking at beta glucose, the hydroxyl group goes up, so it's on top, then it goes down. I'll just change colors. Down, up, then down. So that's a good way to memorize beta glucose. And then we know that alpha glucose has that one difference at your first carbon, where the hydroxyl group is down, and then it just is as follows from beta glucose. So down, up, down. So that's how you can draw either form of glucose. It's really important to note that first carbon though, because that is what distinguishes between them. So as I said, they both have their specific uses, if you like, and we know alpha glucose forms starch in its polymer form, and beta glucose forms your cellulose, so obviously seen in things like plants, and that is due to their unique bonding to that first carbon. Next up, we have fatty acids. So we know all fatty acids have that hydrocarbon chain along with your methyl group and your carboxyl group. So I'll just highlight the methyl group in this instance. And then here is your carboxyl group. So there are three notable types of fatty acid. Firstly, we have our saturated. Oops. Now, the saturated fatty acid structure is as follows. Effectively, you just have that hydrocarbon chain, which I'll highlight in red. So you have a chain of that, and I've just done, for the sake of simplicity, 3, but these can be in much larger length. So saturated compared to the other forms do not have any double bonds. They are all single bonds between your carbons, and then ultimately you have your methyl group and your carboxyl group. Looking at your trans unsaturated fat. Now, these have your double bonded carbon. Note also that they have your methyl group and your carboxyl group. What's unique about your trans unsaturated fats is that they have hydrogen atoms in the upwards direction and then in the downwards direction when we see this double bond. It's important to note as well that although I've only done one double bond in this case, a trans unsaturated fatty acid can have multiple. So to distinguish one, we must just see the hydrogen and how it's in an up and down position. This can be compared to your cis unsaturated fatty acids, which also has that double bond, but has hydrogen atoms both facing that upwards direction. Again, we can see that methyl group and our carboxyl group, but both hydrogens are in the upwards direction for that double bond, which is what gives it this cis unsaturated property. Now, I've made some notes on the side here about the effects of the consumption of these different types of fats. Now, we know that certain types of fats can lead to an increased risk in coronary heart disease. Notably are saturated and trans unsaturated, so the first two that I've highlighted here. Now, these effectively promote low density lipoproteins, which are the storage of cholesterol effectively in the arteries. This, of course, blocks that arterial flow. Now, by comparison, our cis unsaturated fatty acid promotes high density lipoproteins and it regulates low density lipoproteins, therefore making it good. It prevents that buildup of cholesterol in the arteries, which is what occurs with the first two. Therefore, cis unsaturated fatty acids are generally seen to be your good type of fatty acid and not the type that blocks up arteries and can lead to chronic health diseases. So next up, we're going to talk about metabolism, and we've talked about before how all chemical processes maintain life within an organism. I'll talk about the role of enzymes in metabolism. So we know enzymes effectively catalyze metabolism through that binding to a particular reactant. Enzymes lower activation energy, which effectively means that the reaction can go faster, which makes metabolism in turn more efficient. We know with enzymes as well that they are not affected in a reaction. Now, there'll be an upcoming video talking specifically about enzymes. However, we know that they are not used up. So they aren't a reactant. They are just there to catalyze the reaction, making them really important in metabolism. So some quick examples, we know things like pepsin, in the stomach for digestion, or similarly amylase, which we know is in saliva in the mouth, to help digest carbohydrates. Moving on to the specific types of metabolism, we have things like anabolism, which will be our first type, which is the formation of a large molecule from a small molecule with an energy input. So if you look at something like protein synthesis, we know that we're forming that large, complex 3D type protein from a small amino acids. An example of anabolism is condensation, where we have something along the lines of two monomers combining to form a large molecule and another product being water. So this is usually seen when there are an excess amount of hydrogen and oxygen atoms. So we can see in this instance we have a total of, if we just add this up, 1, 2, 3, 4 hydrogens and 2 oxygens. So in this case there would be 2 water molecules formed when these two substances or molecules, if you like, binds together. So this is an example of an anabolic reaction because we have these two simple molecules which bind together to form this one large complex molecule, giving a product as well of 2 water molecules as a consequence of having these additional hydrogen and oxygen atoms in the reaction. Moving on to the second type of metabolism, we have catabolism, which is that large molecule going into the small molecule with that energy source as well. So looking at things like glycolysis, which will be talked about in a later video more specifically, but effectively it's that glucose being broken down into pyruvate and energy, which would typically be like ATP or ATP. So another example is something like hydrolysis. I'm just going to change color here. Hydrolysis, which is effectively the opposite of your condensation. So you can imagine in condensation, we know that water is being a product effectively from that reaction. Hydrolysis is where we add H2O molecules in order to break down this larger molecule. So for instance, if I had this large molecule and then I added water, ultimately it's going to break down back into these two small molecules. So that is effectively what hydrolysis is, and it's an example of catabolism because it's that large to small. Now, as a comparison, we know that this is our catabolism, and this is our anabolism. Catabolism is that large to small. It releases energy in its reaction, and we know it's typically a polymer to a monomer. For instance, we have glycogen, which is that polymer of glucose, being broken down into glucose. So important things to note is that energy and that large molecule to small molecule plus the energy as a product of that as well. Anabolism, is that small to large. It requires energy. It's a monomer to a polymer. So if we're looking at this same example, it would be glucose to glycogen. It needs energy to occur, and as a consequence, a larger, more complex molecule can be synthesized accordingly. A final thing necessary to talk about is urine. Now, I've drawn its structure as follows. And what's interesting about urine is how it can be synthesized both naturally and artificially. So naturally, we see it in the human body in the urine cycle. We know it is released in urine, but we know artificially speaking, it can be produced via cyanic acid and ammonia. What's interesting is this idea of vitalism, which was a theory in the 19th century about how substances produced by living organisms cannot be artificially synthesized. Now, this was falsified through the discovery that urea is effectively cyanic acid and ammonia, and we also know how it can be produced naturally in the body, therefore disproving this idea of vitalism. So that's it for today's video, covering 2.1 syllabus and the idea of metabolism. Thanks for your time, and thanks for watching.